Hey guys, it is, I hope, last day of notes for this unit. So hopefully all goes well. I think it should be not too bad. The hardest stuff I think is from the last video, the incomplete and codominant stuff. So make sure you get a handle on that. So our last type of inheritance are called polygenic traits. And just like the name suggests, poly means many. Genic is obviously referring to genes. So we have many genes that are influencing one trait. Okay, there's kind of a dead giveaway right here. Okay, any trait that has a wide range of phenotypes is going to probably be polygenic. So most of the things we've talked about have been, we would say, okay, possible phenotypes, either tall or short, right? There's two phenotypes. Or maybe with multiple allele traits like blood type, you could have a few phenotypes. You could have A type, B type, AB type, or O, but that's it, right? You just have a handful of phenotypes. In a polygenic trait, you've got lots and lots of phenotypes. So things like skin color, build, I'm talking about how thick are your bones? Are you kind of naturally predetermined to be skinny or have, be obese or whatever, or height. Okay, so imagine, and we're going to exclude people that have genetic disorders affecting height, like dwarfism or gigantism. But if we took, lots and lots of, we'll go with females. Okay, we take like 200 females, their average height is going to be about 5'6". Okay, and then if we went, either way, and then we could go a little further this way if we want, or we can go this way, 5'7", 5'8", 5'9", 5'10", and we could go that way if we wanted to. Okay, and we plotted how many down this way. We would get something that looks like this. Okay, so we'd have lots of women in this kind of mid-range from 5'5" to 5'8" or so, and a few that are taller, and this could extend quite a ways out here, getting lower and lower. Like you can find women that are 6 feet tall. It's not unheard of. 6'1", 6'2", sure. But as you get any taller than that, it gets very, very uncommon. And the same idea going this way, it gets very, very uncommon. So there's not like one or two phenotypes. We can be anywhere in this range. Okay? So we have this wide range of phenotypes, and they usually cluster kind of somewhere in the middle and can go out either way. And so the reason for this is because, again, there are many genes impacting this. And so it becomes a little bit more complicated when we try to think about it a little bit. So, for example, skin color is a classic polygenic trait. And they think there's only maybe like 6 genes, but they, in your book on page 216, they kind of simplified it even to 3 genes, just so you can kind of get an idea of it. Okay, so in their example, there's 3 genes affecting skin color, and for each one, you could have the dominant, which is for dark, or the recessive, which is for light. So if you get the recessive, homozygous recessive for all 3 genes, you're going to be very, very fair. Okay? If for 2 genes you have 2 recessives, but for 1 gene you have the dark, you're going to be a little bit darker. Okay? And then you go all the way to the other range. For all 3 genes, if you have both dominant alleles, okay, you're going to have very dark skin. So we start to see this kind of a range. And again, I think that they've simplified it even still, because I'm pretty sure they have determined that there's 6 genes that determine skin color. So they have this much of a range, but in reality, more than 3 genes, there's like 6 genes. So it can be, You know, there's obviously a very wide range of natural skin color, and then depending on how you treat your skin, whether you allow yourself to be out in the sun or wear sunscreen or whatever, then we can have everything in between. So, again, polygenic, lots and lots of possibilities there. Then we have sex-linked traits. And what makes something sex-linked is the gene is on a sex chromosome. Okay, either the X chromosome or the Y chromosome. Okay, but when we're talking about sex-linked traits, the X chromosome, I think we kind of mentioned, is very, very, very much larger than the Y chromosome. It has a lot more genes. So most sex-linked traits are X-linked traits just because the Y has hardly any genes on it. And so males have only one. They have a single X chromosome. Remember, their other sex chromosome is a Y. So if that chromosome, their one X chromosome, has the allele for some kind of a disorder, they don't really have a chance to have a backup X. Okay? Females have two X's. So if one of those X's has the allele for the disorder, they can have basically what I like to call a backup with a functional copy of the gene. And I say, let's explore this. Okay, let's talk about Roxana Fee's family. So one classic example is red-green color blindness. Okay, so here's my grandpa Wally, and here's my grandma Murdy, and grandpa is colorblind, so we're going to fill him in. Okay, so boxes are for males, circles are for females, and we're going to say fill in means they have a disorder. Half means a carrier. Empty, no bad alleles. Okay, so grandpa has color blindness, grandma Murdy normal. Okay, they have 4 kids. They have 3 girls and 1 boy. Okay, so grandpa must have X, and I'm just going to put a little dot there to show that that's the one with the disorder, Y. And grandma has 2 normal X's. All right, so the 3 girls, okay, so they had to get an X from each parent, right? So they got an X from grandpa and an X from grandma. This is my mom, Linda. So she got this X from grandpa, and she got one of these X's from grandma, doesn't really matter. Okay, would she have color blindness? Okay, no. She has this backup X that doesn't have anything wrong with it, right? But, so she would have normal vision, okay, but she has this kind of a tainted X that has the color blindness allele that she could pass on. So I'm going to fill that one in halfway. Okay, so normal vision, but has one of these X's for color blindness. Okay, here's my Aunt Lori. Okay, so same idea. She could only have got this X from grandpa. So she must also be a carrier. And my Aunt Christine, Chris, same. So all the daughters would have to be carriers. Okay, how about my Uncle Scott here? Okay, so he's a male, so he's XY. So he got this Y from grandpa. That's what makes him a boy. And one of these normal X's. So would he be colorblind? No, he would not be colorblind, right? Because he has no defective X's. So we don't put anything in there. He has no bad alleles to be had. Okay, so here comes my dad Kermit, my Uncle Jay, my Uncle Ken, and my Aunt Melissa. Okay, so they're married now. All right, 4 kids. 1, 2, 3, 4. 3 kids. 1, 2, 3. 2 kids. And 2 kids. Okay, boy, girl, boy, girl. That's a box. Here's all my cousins on my mom's side of the family. Do you feel like you know me now? So, think about this, okay? So here's me. Here's Miss Fees. Okay, just using my genetics. Okay, I didn't fill in my dad, so he's not colorblind. Is it possible that I am colorblind? Okay, no, because my dad did not have a defective X to give me, right? If he did, he would be colorblind, and I would probably be considered a carrier. Okay, might I be a carrier? Okay, yes, I could be a carrier, because I could have got this X from mom that has that color blindness gene, or I could have got this X from mom. I don't know. Okay, the only way I'll really know is if I someday have sons or something and one of them is colorblind. And if any of my sons are colorblind, I'll know I'm a carrier. So I'm going to just leave myself here a question mark because I don't know. Okay. I'm pretty sure, it's kind of weird that I don't know, but I'm pretty sure Jesse's colorblind. That's my brother. And Allison, again, we wouldn't know. Okay. I know this one's colorblind, and I'm pretty sure this one's colorblind. And if you look at the boys, so girls would all be question marks, don't know, except for this one, because parents, neither one, unless she's a carrier, we don't know, I guess, but she's probably normal. He doesn't have color blindness. Why is it more common in males, right? They only need the one bad X for this to happen. So hopefully that kind of makes sense. Usually we'll see X-linked traits show up in males because they only have one X, right? All right, so then we have something kind of different, sex-influenced genes. Okay. So these can be expressed in either gender, but are influenced by the male or female hormones that are floating around in somebody's body. So a really classic example, if you're a guy and you're like, oh, I wonder if I'll go bald, right? You hear a lot of people say, well, look at your mom's dad and see what his hair did, and that's, a good indicator. And that's a little bit of an oversimplification, but notice they didn't say, well, look at your mom, right? They said, look at your mom's dad, because your mom would not have necessarily the same hormones to show if she would go bald genetically. Okay, so male pattern baldness is a sex-influenced gene. Two people could have the same genetics for going bald, right? But if one is a male, the hormones in their body are going to kind of influence how that phenotype is shown. But the hormones in males can make the expression of the phenotype baldness more common. Again, even if the genetics, the genotype is the same, the gender can influence whether the phenotype is fully expressed or not. All right. And then we have, what can we do about it? Well, something. Okay, so environmental impact on phenotype. So what your body takes in, or its diet, how much you're exposed to various mutagens like sunlight, and more can affect traits in humans. But in some organisms, gender has an even more profound impact. There's some species of like butterfly where if they're incubated at one temperature, most of them become males, and if they're incubated at a different temperature, they become females. Or, the patterns of things, like the swirls on turtle shells, I think can be influenced by their environment when they're still in the egg. And I just had another example here. Honeybee queens, okay, so remember queens are like 2 times bigger than all the other bees, at least, and they produce a massive amount of egg, and all the other females don't make any eggs. Okay, so their phenotype, their appearance is very, very different. Okay, but they're genetically no different than all the worker bees. Okay, so what makes them different? Well, it is the kind of nutrients that is in the food they are given when they are not even adult yet, but just larvae. Okay, changes how they develop. So environmental impact can change the phenotype, the physical trait of an organism a lot. All right, and so here's another kind of tack onto that idea. Remember, genetics predicts probabilities. That's a B. Not certainties. Okay, so maybe in your genetics, you're one of those crazy lucky people who genetically you're very likely to be thin, right? So it seems like your mom and dad are thin. They don't have to work at it. They're just naturally thin people. But if you choose to do nothing and be a lump on your couch for 10 hours a day and never exercise and eat a bunch of potato chips and cheese, you probably won't be, right? Just because your probability of being thin genetically is high doesn't mean your environment and behavior can't override that. All right. This last part I probably wouldn't have thought was too important, but it was on the WNCC syllabus, so here we are. So geneticists study a wide range of organisms, and the things that makes an organism really good for genetic study are going to be, number one, it's small. Okay, small organisms are easy to manage. They're easy to, keep in containers in the lab and cross this male with this female. It's a little easy. They have a short lifespan. Okay, so if I want to see the result of a genetic cross, I would like not to have to use something that can't reproduce for the first 10 years of its life, and gestation lasts a year, and then, so that would take forever, right? So I want something with a short lifespan, something that can be born, reproduce, we can see the results of its reproduction in a short amount of time, and also have easily identifiable traits. So Gregor Mendel picked a pretty good one, peas. So they're fairly small, right? You can keep lots of them in a fairly small amount of space. They have a short lifespan. You could probably do a couple generations in a summer if you were really on the ball and planted things first thing in the spring. And they do have easily identifiable traits, right? So they had purple flowers and white flowers, or tall or short, or the different pea colors and pod shapes and all kinds of things. He picked them mostly because they had a function for his monastery, right? So they could eat them. Okay, they did take a while to grow. Okay, 3 generations in a year is probably not ideal if you're really trying to make some studies. Geneticists around the world have kind of settled on their model organism, and this is the scientific name, and it's the common fruit fly. And if you ever want to get me fired up, start me talking about fruit flies, because I did an entire summer doing genetics research on fruit flies, and some people would love it. I didn't love it. It was not fun for me, because I had to go into the lab every, I think it was 8 hours for like a week at a time, and that includes in the middle of the night. So I would go in on a certain, it was 8 hours because it was 3 times a day. So I'd go in at like 8 in the morning, 4 in the afternoon, and then at midnight. Okay, I have to go into the lab 3 times a day at 8 and 4 and midnight and see how many flies were dying and suck them out of the cage with a tube with the suction power of my mouth, and it sucked. But people like it. They're a great organism because they can be grown in small vials, right? You could have tons and tons of flies in a vial, like a test tube size vial, and you could have like dozens or 100 living in there. They can be born and mate in a very small amount of time. A few days from birth to next generation is about a week. Okay, and they have a bunch of easily studied traits like eye color and wing length, and there's all kinds of things that are fascinating about fruit flies. I'm more of a theory person than an actually do it person. All right, V. Okay, so we're spent all this time talking about probabilities. But just remember that your real world results are probably not exactly going to match your probabilities. Okay, so we can take statistics, and we're not going to go into the statistics of it beyond just to kind of talk about this. We use them to take the probabilities and see if the results agree or not with a certain degree of certainty. Okay, so what does that mean? So if I want to say maybe my Punnett square said some phenotype was 75%, and I did a cross of some organisms and I got 85%. Does that mean everything I did was wrong and I definitely screwed up? Okay, so how do we know? Like, how sure do I want to be that I didn't screw up? And what amount of variance from the probability is reasonable? Okay, and it depends on a couple things. It depends on your sample size. How many offspring did you create? Right? If it's a small sample size, then you can really always not, I'm going to say double negative here. You can never be as sure of your results if you have a small sample size, because random chance can just play a bigger factor here. And also how certain you want to be. Okay, so in good scientific studies, they usually set their like certainness at like 95%, and some studies even set it to like 99%. And this, again, you can take an entire semester of this in college. I would encourage you to. It's a fascinating math course without doing too much, complicated stuff beyond like Algebra 1 level. Actual math functioning. W is going to be, that's a W, genetic engineering. Okay, any process that uses science to change the genome of an organism. Remember, the genome is all of the genes, all of the alleles that make an organism function. So, how can we engineer things genetically? Well, there's the slow path and the fast path. Okay, the slow path is selective breeding. Also known as artificial selection. Okay, where humans just choose parents of the next generation that have the traits that they like. So they're just kind of saying, okay, I want, a dog that is brown and nice. So they'll take two dogs they can find that are brown and nice and cross them together and hope all their puppies are brown and nice, right? So they have some sort of desired traits. And if we want to see big time changes in an organism, we'd have to do this, generation after generation. But pretty much everything that we eat does not happen naturally, right? So any of your fruits and vegetables that you're like, oh, I'm eating natural, well, only kind of, right? Those plants did not exist naturally. Humans created them by crossing individuals with traits that they liked for hundreds and hundreds of years. And then here's the fast way, is we can genetically modified organisms. We, through very complicated science, take genes from one species and add it to a different species genome. Okay, so classic example of this, people that have some kinds of diabetes are unable to make enough of a certain protein called insulin, which you have probably heard of. So when they first figured out what diabetes was, they're like, okay, well, we can get insulin from certain organs of animals when we butcher them. So they're getting insulin from cattle, they're getting insulin from pigs, but it was, kind of a not handy process. Okay, so they're like, okay, well, we can take the gene for insulin and put it into bacteria. So now remember, genes are instructions for making proteins. So this bacteria now has instructions for making insulin. So it's going to follow those instructions. It's going to make insulin. And we can produce bacteria in large quantities very, very easily. And so the supply of insulin for diabetics became much more consistent and affordable after they figured out how to genetically modify organisms. Any crops that you plant that say Roundup Ready, those would also be for sure genetically modified. They've had a gene put into their genome that allows them to break down Roundup so it doesn't kill them, but it only kills other organisms. So that's the end of this unit. I'll try to get some review materials up shortly. Well, you're going to get this on Tuesday. We're probably looking at a test maybe next Monday. So I'll get more information out about that soon. See you later.